This video will explain different immunologic and serologic tests that can be performed in a medical laboratory. The goal of the video is to be able to explain how antigens and antibodies can be used as laboratory tools. The first technique we'll talk about today is agglutination. And in agglutination, you have the clumping of an insoluble particle with an antibody. They bind together and they form a large clump or agglutinate. And what will happen is the antibodies, when they are in contact with their specific antigen, will bind. And if you have an antigen which has many different epitopes on it, you will have cross-linking occurring, as you can see here. So you have one antibody binding to an epitope on more than one particle, insoluble particle. Because they cross-link, they form what's known as a lattice, which is the structure you see to the right, basically a network of antibody and antigen all bound together. Now this lattice is relatively heavy, so the bigger it becomes, the more likely it is to fall out of the solution due to its weight. That falling out of solution is the agglutination process occurring, the clumping occurring. In a direct agglutination test, usually the presence of a clump means a positive result. Now there's different types of insoluble particles we can use in an agglutination reaction. One easy one is if you're looking for the presence of a particular antibody to a bacterial cell, you can use the bacterial cell itself. You can use a latex particle where you take an antigen from another organism and attach it to the outside of the particle and use that as your agglutinant. And then most commonly in blood banking we see hemagglutination, where you have the attachment of antibodies to red blood cells forming a nice big clump. Agglutination inhibition is similar to agglutination, however there are some key differences you need to understand. First off, agglutination inhibition is competitive. That means you're going to have two antigens vying for binding of the same antibody. And so you're going to see a little bit of a different reaction occurring because of that. And also, in the end, no clumping is actually a positive result. And you'll see why that might be as we go through what happens in an agglutination inhibition reaction. So you start your reaction with a reagent or a commercial antibody that is particular for the antigen of interest that you're looking for in your patient. And you'll have that antibody, and to it you will add the patient specimen. It may or may not have antigen, but in this case we're going to see what happens when the patient specimen has antigen in it. So if you add the patient sample with the antigen in it, what it will do is bind to the commercial antibody that you had in the first step. This will bind up all of the free antibody, and there will be none left to react with anything else. So the step after this is when you add another commercial product. In this case you have an insoluble particle that's coated with the same antigen that you're looking for. And if you added it to a tube of unbound antibody, those antibodies would bind to this commercial particle and cause agglutination because the particle is insoluble. But in this case, because all of the antibody has been bound by the antigen that came from the patient sample, you won't see any clumping because there's no free antibody left to bind the second reagent and form a lattice. So no clumping in this case is a positive reaction, and that means that the patient does have the antigen that we were looking for, the antigen of interest. If they didn't have it, this antibody would be free to bind the commercial antigen added in step 2, and in step 3 you would have seen visible clumping. Precipitation reactions use the same principle as agglutination, except for the following things. First off, in precipitation, the antigen is very small, and because it's really small, that means it's pretty soluble in the solution. This could be something like a toxin, a poison, a really small protein that you're looking for. And since it is small and soluble, the lattice that's formed is also small and very hard to see. You can see it. Oftentimes a precipitation reaction will be run in a gel and it makes it a little bit easier to visualize, but sometimes what will happen is the test that is normally a precipitation reaction will be converted into an agglutination reaction. And that is done by attaching the antigen that you're looking for, or the antigen that you are looking for antibodies to a latex particle. So if this is the antigen that you have and you want to determine if a patient has the antibody to it, what you'll do is you'll take this antigen and you'll attach it to a big latex particle. And then you'll mix those latex particles with the antibody, or the serum in this case, of the patient who you suspect might have the antibody. And if they have it, they will bind to the antigen on the latex particle and cause agglutination. So it takes a hard-to-read precipitation reaction and converts it to an agglutination reaction, and the principle is pretty much the same. Hemolysis can also be used as a positive reaction that indicates the presence of a particular antibody, and that's because binding of antibody activates a series of proteins known as complement proteins. And what those proteins do is they cause the destruction of cells, the lysis of certain cells. They form a hole in the cell membrane which causes the cell to be permeable. Water will rush into the cell and it will cause it to burst. And this complement can be activated through the binding of antibody to its antigen. So if you have a red blood cell, for example, that has antibody bound to it, one of the things that can happen is if this is in vivo, or if this is in a tube that has serum or plasma as well in it, where the complement proteins would be present, the complement proteins will recognize the binding of the antibodies to the red cell, and that initiates a very complex cascade of proteins that in the end will result in the formation of little holes or pores in the surface, or the cell membrane surface. Those holes will essentially cause water to rush in, as I said, and cause the red blood cell to burst. So in that case, hemolysis, or destruction of a red cell, indicated that there were antibodies present and that the complement caused the destruction of that cell as a result. A little bit fancier than the techniques we've talked about before is the fluorescent antibody technique. And in this case, what you do is you conjugate an antibody specific for an antigen to a fluorochrome, or something that will give off light under a certain wavelength of light. Oftentimes the fluorochrome is fluorescein. It's a very commonly used fluorochrome for whatever purpose, for whatever reason. And what will happen is this fluorescent antibody will be added to the sample, and if the antigen is present, it will bind. And then what you have to do is you have to have a special machine or microscope that is capable of detecting the fluorescence in order to tell you how much binding has occurred, if it has occurred at all. So if you have a liquid sample, what you can do is you can actually put the cells that are bound or unbound through a machine that will, or a cell sorter that will count every fluorescently labeled particle that goes through it. And so you can get a quantitative analysis of how much of the antigen you've got. Or if you have a solid sample, let's say you have a tissue that you want to look and see where a particular antigen might be in it, you can prepare that tissue on a slide and use a fluorescent microscope in order to determine where the antigen is, and then get a rough idea of how much based on how much fluorescence is given off. As you can imagine, this is a little bit pricier because you have to have the creation of those fluorescent antibodies, but it can be very specific and very useful in the laboratory and in research laboratories as well. For the last two topics, the enzyme immunoassay and Western blot, I'd like you to view two animations that I've placed in the course modules and kind of see how they work through those animations. Make sure you pay attention to the difference between a direct and indirect enzyme immunoassay, and look at how the antibodies are used specifically in the Western blot. We'll talk about both of them in class. If you have any questions, feel free to let me know.